Hello and good morning, everyone. I am Celine Zaire V. Salem, and I am here today with my partner, Miss Kyle Jean Regidor. And together, we will discuss to you the Chapter 2 Biological Foundations Heredity, Prenatal Development, and Birth. But before we begin, let us participate in a short meditation first. Find a comfortable place. Now slowly close your eyes and appreciate every sensation in your body. Allow thoughts to come to you and breathe. Breathe in, breathe out. Breathe in, breathe out. Slowly breathe in and out. Repeat a short mantra with me. Healthy body, calm mind, compassionate heart. Healthy body, calm mind, compassionate heart. Now slowly open your eyes. Okay, so I hope everyone was relaxed. So now we can proceed with the objectives for Chapter 2. The objectives are to learn about inherited factors that affect development, to learn about the timetable that governs before birth, and along the way, get answers to common questions about pregnancy, to discuss some of the problems that occur during the development before birth, and to find out how an expectant mother can prepare for birth and what labor is like. If you ask your parents to name the most memorable experience of their lives, many immediately mention the events associated with the birth of their children, from the initial exciting news that a woman is pregnant through birth 9 months later. Pregnancy and birth evoke awe and wonder. The period before birth is the foundation for all human development and is the focus of this chapter. Pregnancy or conception begins when egg and sperm cells unite and exchange hereditary material. Each egg and sperm cell has 23 chromosomes. Chromosomes are thread-like structures in the nucleus that contain genetic material. When a sperm penetrates an egg, their chromosomes combine to produce 23 pairs of chromosomes. The first 22 pairs of chromosomes are called autosomes. The 23rd pair determines the sex of the child, so these are known as sex chromosomes. It can be X and X for a girl and X and Y if it's a boy. An example of a hereditary disease is sickle cell disease. It is characterized by red blood cells that are long and curved like a sickle. These stiff, misshapen cells cannot pass through small capillaries, so oxygen cannot reach all parts of the body. About 10% of people with the disease die by the age 20, and 50% die by the age of 50. Each chromosome consists of one molecule of deoxyribonucleic acid, or DNA for short. To understand the structure of DNA, imagine four different colors of beads placed on two strings. The strings complement each other precisely. Wherever a red bead appears on one string, a blue bead appears on the other. And wherever a green bead appears on one string, a yellow one appears on the other. DNA is organized that way, except that the four colors of beads are four different chemical compounds, namely adenine, thymine, guanine, and cytosine. The strings, which are made up of phosphates and sugars, wrap around each other, creating a double helix as shown in the figure. The order in which the chemical compound or beads appear is a code that causes the cell to create a specific amino acid, protein, or enzyme, which are biological building blocks. Each group of compounds that provides a specific set of biochemical instructions is a gene. In the Human Genome Project during 2013, a person has 46 chromosomes, include roughly 20,500 genes. Chromosome 1 has the most genes, which is nearly 3,000, and the Y chromosomes has the fewest, just over 200. Now let's review these few scientific terms. Can you remember all of these? A genotype is a complete set of genes that make up a person's heredity, while phenotype is the physical, behavioral, and psychological features that result from the interaction between one's genes and the environment. Alleles are the variations of genes. Homozygous means when the alleles in a pair of chromosomes are the same, and for heterozygous it means when the alleles in a pair of chromosomes differ from each other. Dominant means form of an allele whose chemical instructions are followed, while recessive is when allele whose instructions are ignored in the presence of a dominant allele. Incomplete dominance is when one allele does not dominate another completely. The simple genetic mechanism responsible for sickle cell disease, which involves a single gene pair with one dominant allele and one recessive allele, is also responsible for numerous other common traits as shown in the table. For the dominant phenotype, we have curly hair, normal hair, dark hair, thick lips, cheek dimples, normal hearing, normal vision, farsightedness, normal color vision, type A blood, type B blood, or H positive blood. And for the recessive phenotype, we have straight hair, patterned baldness in men, blonde hair, thin lips, no dimples, some type of deafness, nearsightedness, normal vision, red-green color blindness, type O blood, and Rh negative blood. In each of these instances, individuals with the recessive phenotype have two recessive alleles, one from each parent. Individuals with the dominant phenotype have at least one dominant allele. Most of these traits listed in the table are biological and medical phenotypes. The same genetic mechanism can cause serious disorders. Genetic disorders can be developed in two ways. First, it can be inherited. Second, it can be caused due to more or fewer chromosomes. Example of inherited disorders in relation to recessive alleles are the sickle cell disease and the PKU or the phenylketonuria. PKU is characterized by the lack of important liver enzyme that converts phenylalanine into tyrosine. According to Diamond et al., if phenylalanine is not converted, it becomes poison, which then causes mental retardation. Another inherited disorder, which is in relation to a dominant allele, is the Huntington's disease. It is characterized by a fatal progressive degeneration of the nervous system. There are only a few serious disorders caused by dominant alleles. Why? If the allele for the disorder is dominant, every person with at least one of these alleles would have the disorder. Individuals affected with these disorders typically do not live long enough to reproduce. So, dominant alleles that produce fatal disorders soon vanish. Now let's move on to the abnormal chromosomes. Sometimes individuals do not receive the normal complement of 46 chromosomes. If they are born with extra, missing, or damaged chromosomes, development is disturbed. The best example is Down syndrome. People with Down syndrome have almond-shaped eyes and a fold over the eyelid. Their head, neck, and nose are usually smaller than normal. By childhood, most aspects of cognitive and social development are seriously retarded. What causes Down syndrome? Individuals with Down syndrome typically have an extra 21st chromosome that is usually provided by the egg. Why the mother provides two 21st chromosomes is unknown. However, the odds that a woman will bear a child with Down syndrome increase markedly as she gets older. Why? A woman's eggs have been in her ovaries since her own prenatal development. Eggs may deteriorate over time as part of aging or because an older woman has a longer history of exposure to hazards in the environment that may damage her eggs, such as X-rays. Abnormal sex chromosomes can also disrupt development. Notice that no disorders consist solely of Y chromosomes. The presence of an X chromosome appears to be necessary for life. As shown in the table, here are the list of four more frequent disorders associated with atypical numbers of X and Y chromosomes. We have the Klinefelter syndrome, the XYY complement, Turner syndrome, and XXX syndrome. That we understand the basic mechanisms of heredity, we can learn how heredity and environment work together to produce behavioral and psychological development. Many people mistakenly view heredity as a set of phenotypes unfolding automatically from the genotypes that are set at conception. Nothing could be further from the truth. Although genotypes are fixed when the sperm fertilizes the egg, phenotypes are not. Instead, phenotypes depend on both genotypes and on the environment in which individuals develop. To begin the discussion, we need to look first at the methods that developmental scientists use. First is the behavioral genetics. Behavioral genetics is the branch of genetics that deals with inheritance of behavioral and psychological traits. It is complex in part because behavioral and psychological phenotypes are complex. Another is the polygenic inheritance, which are the result of the combined activity of many separate genes. Scientists also study twins. Twins and adopted children provide some important clues to the role of heredity. In twin studies, researchers compare identical and fraternal twins. Identical twins are called monozygotic twins because they come from a single fertilized egg that splits in two. Because identical twins come from the same fertilized egg, the same genes control their body structure, height, and facial features, which explains why identical twins look alike. In contrast, fraternal or dizygotic twins come from two separate eggs fertilized by two separate sperms. Adopted children are another important source of information about heredity. They are compared with their biological parents who provide the children's genes and their adoptive parents who provide the children's environment. If an adopted child's behavior resembles that of his or her biological parents, this shows the impact of heredity. If the adopted child's behavior resembles his or her adoptive parents, this shows the influence of the environment. These and other methods are not foolproof. A potential flaw in twin studies is that parents and others may treat monozygotic twins more similarly than they treat dizygotic twins. This would make monozygotic twins more similar than dizygotic twins in their experiences as well as their genes. Next is paths from genes to behavior. Do you remember your younger years when you would still go out and play with your friends? When you would go to school and excitedly raise your hand to answer your teacher's question? Or are you the one to shy away when the teacher asks? We will cover that in this part. Number one, heredity and environment interact dynamically throughout development. A traditional but simple-minded view of heredity and environment is that heredity provides the clay of life, and experience does the sculpting. In fact, genes and environments constantly interact to produce phenotypes throughout a person's development. A good example of this genotype-phenotype link is seen in the disease PKU. PKU is a homozygous recessive trait in which phenylalanine accumulates in the child's body, damaging the nervous system and leading to a retarded mental development. Phenylalanine is abundant in many foods that most children eat regularly, like meat, chicken, eggs, cheese, so that the environment usually provides the input phenylalanine necessary for the PKU to emerge. However, in the middle of the 20th century, the biochemical basis for PKU was discovered, and now newborns are tested for this disorder. Infants who have genotype for the disease are immediately placed on a diet that limits phenylalanine, and the disease does not appear. We used a rare disease (PKU) and once in a lifetime event to show intimate connections between nature and nurture in human development. These examples may make it seem as if such connections are rare, but nothing could be further from the truth. At a biological level, genes always operate in a cellular environment. Genetic instructions interact constantly with the nature of the immediate cellular environment, which can be influenced by a host of much broader environmental factors, like hormones triggered by a child's experience. This continuous interplay between genes and multiple levels of environment that drives development is known as epigenesis. Because of the epigenetic principle that you need to be wary when you read statements such as X% of a trait is due to heredity. In fact, geneticists often use correlations from twin and adoption studies to calculate a heritability coefficient, which estimates the extent to which differences from people reflect heredity. For example, intelligence has a heritability coefficient about zero point 0.5, which means that about 50% of the differences in intelligence between people are due to heredity. Why be cautious? One reason is that many people mistakenly interpret heritability coefficients to mean that 50% of an individual's intelligence is due to heredity. This is incorrect because heritability coefficients apply to groups of people, not to a single person. Second reason for caution is that heritability coefficients apply only to a specific group of people living in a specific environment. They cannot be applied to other groups of people living in the same environment or to the same people living elsewhere. Number two, genes can influence the kind of environment to which a person is exposed. In other words, nature can help determine the kind of nurturing that a child receives. For example, a child may receive plenty of attention and encouragement from teachers. In contrast, someone who is not as bright and is more withdrawn, again due in part to heredity, may be easily overlooked by teachers. In addition, as children grow and become more independent, they actively seek environments that fit their genetic makeup. Children who are bright may actively seek peers, adults, and activities that strengthen their intellectual development. Similarly, people who are outgoing may seek the company of other people, particularly extroverts like themselves. This process of deliberately seeking environments that fit one's heredity is called niche picking. It is first seen in childhood and becomes more common as children grow older and can control their environments. Number three, environmental influences typically make children within a family different. Some parenting practices are thought to be more effective than others, and parents who use these effective practices are believed to have children who are, on average, better off than children of parents who don't use these practices. This view leads to a simple prediction. Children within a family should be similar because they all receive the same type of effective or ineffective parenting. However, dozens of behavioral genetic studies show that in reality, siblings are not very much alike in their cognitive and social development. Does this mean that family environment is not important? No. These findings point the importance of non-shared environmental influences, the forces within a family that make children different from another. Each child is likely to have different experiences in daily family life. For example, parents may be more affectionate with one child than another. They may use more physical punishment with one child than another, or they may have higher expectations for school achievement by one child than another. All these contrasting parental influences tend to make siblings different. The many changes that transform an egg fertilized by a sperm cell into a newborn human constitute prenatal development. There are three periods: the period of the zygote, the period of the embryo, and the period of the fetus. For the period of the zygote, weeks 1 to 2, after the fertilization, it creates the zygote. Another form of fertilization is the artificial means, which is in vitro fertilization. It is a process by which sperm and egg are mixed in a petri dish to create a zygote, which is then placed in a human's uterus. For couples who are known to be risk for genetic disorders, usually use the pre-implantation genetic screening or PGS. Another effort to improve human species is called eugenics. It's when people do effort by allowing only certain people to mate and pass along their genes. By the end of the first week, the zygote reaches the uterus. The next step is implantation, which the zygote burrows into the uterine wall and establishes connections with the woman's blood vessels. The implanted zygote is less than a millimeter in diameter, yet its cells have already begun to differentiate. A small cluster of cells near the center of the zygote, the germ disk, eventually develop into the baby. The layer of cells closest to the uterus becomes the placenta, a structure through which nutrients and wastes are exchanged between the mother and the developing organism. Next is the period of the embryo at weeks 3 to 8. After the zygote is completely embedded in the uterine wall, it is called an embryo. It is important to remember that during the period of the embryo, body structures and internal organs develop. At the beginning of this period, three layers form in the embryo. The outer layer develops into ectoderm. It becomes hair, the outer layer of the skin, and the nervous system. The middle layer, or mesoderm, forms muscles, bones, and the circulatory system. And the inner layer, or endoderm, forms the digestive system and the lungs. The embryo rests in a sac called the amnion, which is filled with amniotic fluid that cushions the embryo and maintains a constant temperature. The embryo is linked to the mother via two structures, the placenta and the umbilical cord. The umbilical cord houses blood vessels that join the embryo to the placenta. In the placenta, the blood vessels from the umbilical cord run close to the mother's blood vessels, allowing nutrients, oxygen, vitamins, and waste products to be exchanged between mother and embryo. Growth in the period of the embryo follows two important principles. First, the head develops before the rest of the body. Such growth, from the head to the base of the spine, illustrates the cephalocaudal principle. Second, arms and legs develop before hands and feet. Growth of parts near the center of the body before those that are more distant illustrates the proximodistal principle. Growth after birth also follows these principles. Period of the fetus weeks 9 to 38. The final and longest phase of prenatal development is called the period of the fetus. It begins at the ninth week and ends at birth. The increase in size of the baby to be is remarkable. At the beginning of this period, the fetus weighs less than an ounce. About 4 months, the fetus weighs roughly 4 to 8 ounces, which is large enough for the mother to feel its movements. And at the last 5 months of pregnancy, the fetus gains additional 7 or 8 pounds before birth. During the fetal period, the finishing touches are placed on the many systems essential to human life, such as respiration, digestion, and vision. Some of the highlights include the formation of cells curls to form a tube. This becomes the brain of the fetus. During the period of the fetus, all regions of the brain grow, particularly the cerebral cortex. It is the wrinkled surface of the brain that regulates many important human behaviors. Another is the formation of the penis and scrotum, but in female fetus, this hormone is absent, so the same cells become a vagina and labia. Also, the skin thickens and is covered with a thick greasy substance or vernix that protects the fetus during its long bath in the amniotic fluid. The fetus responds to sound at 29 weeks after conception and by 34 weeks can distinguish different speech sounds, including different vowel sounds and different languages. The fetus can learn and remember. When a fetus is exposed repeatedly to distinct sounds, like the mother's voice, or distinct tastes, like what the mother eats, the fetus recognizes those stimuli after birth. With these and other rapid changes, by 22 to 28 weeks, most systems function well enough that the fetus born at this time has a chance to survive, which is why this age range is called the age of viability. By this age, the fetus has a distinctly baby-like look, but babies born this early have trouble breathing because their lungs are not yet mature. Also, they cannot regulate their body temperature very well. At this point, I hope everyone has retained some of the informations that were discussed. So moving on, we have the influences on prenatal development. The general risk factors include nutrition. The mother is developing child's sole source of nutrition, so a balanced diet that includes foods from each of the five major groups is vital. Mothers need to consume key nutrients such as folic acid, iron, and calcium. A diet that lacks these nutrients can harm the developing child. For example, if mothers do not consume adequate amounts of folic acid, their babies are at risk for spina bifida. It is a disorder in which the embryo's neural tube does not close properly. When the neural tube does not close properly, the result is permanent damage to the spinal cord and the nervous system. Consequently, many children with spina bifida use crutches, braces, or wheelchairs. I have a question. Does a pregnant woman's mood affect the zygote, embryo, or fetus in her uterus? Is a woman who is happy during pregnancy more likely to give birth to a happy baby? These questions address the impact on prenatal development of chronic stress, which refers to a person's physical or psychological responses to threatening or challenging situations. According to Steneva et al., women who report greater anxiety during pregnancy more often give birth early or have babies who weigh less than average. According to Grizenko et al., what's more, when pregnant women are anxious, their children are less able to pay attention and more prone to behavioral problems as preschoolers. At this point, my partner, Miss Kyle Jean Rehidor, is supposedly the one to discuss to you this until the end of chapter 2. However, she is experiencing some health problems, and so please bear with me as I will try to go over her part quickly. Mother's age. For teenage girls, there is a greater risk in giving birth early, and their children often have behavioral problems. In 20s, it is considered the prime childbearing years, and for the 30s and older, it is less fit for pregnancy and is considered dangerous for women. Prenatal development, infancy, and early childhood. In the late 1950s, teratogen drug was discovered. Teratogen is an agent that causes abnormal prenatal development. An example of teratogen is thalidomide. It is a powerful teratogen, an agent that causes abnormal development. Ultimately, more than 7,000 babies in the world were harmed before thalidomide was withdrawn from the market. Prompted by the thalidomide disaster, scientists began to study teratogens extensively, and today we know a great deal about teratogens. Most teratogens fall into one of the three categories: either drugs, diseases, or environmental hazards. Examples of teratogenic drugs and their consequences are: for the alcohol, the potential consequences are fetal alcohol syndrome, cognitive deficits, heart damage, and retarded growth. Aspirin can cause two deficits in intelligence, attention, and motor skills. Caffeine can lower birth weight and decrease muscle tone. Cocaine and heroin can cause retarded growth and irritability in newborns. Marijuana can cause lower birth weight and less motor control, and nicotine can cause retarded growth and possible cognitive impairments. Teratogenic diseases and their consequences. Examples are AIDS, and the potential consequence is frequent infections, neurological disorders, and death. Chlamydia can cause premature birth, low birth rate, or eye inflammation. Chickenpox can cause spontaneous abortion, developmental delays, or mental retardation. Cytomegalovirus can cause deafness, blindness, abnormally small head, and mental retardation. Genital herpes can cause encephalitis, enlarged spleen, improper blood clotting, and rubella or German measles. It can cause mental retardation, damage to eyes, ears, and heart. Syphilis can cause damage to the central nervous system, teeth, and bones. And toxoplasmosis can cause damage to the eyes and brain and learning disabilities. For environmental teratogens and their consequences, we have air pollution. It can cause low birth weight, premature birth, and lower test scores. Another is lead. It can cause mental retardation. Mercury can cause retarded growth, mental retardation, and cerebral palsy. PCBs can cause impaired memory and verbal skills. And X-rays can cause retarded growth, leukemia, and mental retardation. Let's take a look at the figure 2.8 to see how teratogens influence prenatal development. If you see the orange and gold lines with the labels on the end, it is the major structural abnormalities and physiological defects and minor structural abnormalities that happens caused by teratogens on the developing baby. So as we can see, in the first and second week of the developing baby, usually it is not susceptible to teratogens. However, it can cause prenatal death. In the central nervous system, teratogens affect the fetus from the third week to the 16th week. And then physiological defects and minor structural abnormalities happen from 20 to 38 weeks. For the heart, we have major structural abnormalities from the third week to the sixth week, and physiological defects and minor structural abnormalities from the 6th week to 8th week. And for the arms, we have from 4th week to 6th week, and then for the physiological defects, we have 7th to 8th. For the eyes, it starts around the 4th week to around the 8th week. And then the physiological defects start at the 8th week to the 38th week. And for the legs, it starts around the 4th week and then to the 7th week. And then the physiological defect starts at the 7th week to the 8th. For the teeth, it starts around 6th week and then to the 8th week. And then the physiological defect starts at 9th week to the 38th week. For the palate, it starts around the 6th week and then to the 9th week. Physiological defects also start at 9th week and stops there. For the external genitalia, it starts around the 7th week and to the 9th week, and then the physiological defect starts at 9th week to the 38th week. And lastly, for the ear, it starts around the 4th week to the 9th week, and then the physiological defect starts at 9th week to the early days of 20th week. There are 5 important general principles about how teratogens affect or influence prenatal development. Number 1. The impact of a teratogen depends on the genotype of the organism. Apparently, heredity makes some individuals more susceptible to teratogen. 2. The impact of teratogens changes over the course of prenatal development, as shown from the last figure, figure 2.8. Consequences of teratogens differ from the periods of the zygote, embryo, and fetus. Number 3. Each teratogen affects a specific aspect or aspects of prenatal development. Teratogens do not harm all body systems. Instead, damage is selective. When women contract rubella, their babies often have problems with their eyes, ears, and heart, but have normal limbs. When mothers consume PCB-contaminated fish, their babies typically have normal body parts and normal motor skills, but below average verbal and memory skills. 4. The impact of teratogens depends on the dose. Just a single drop of oil won't pollute a lake. Small doses of teratogens may not harm the fetus. And last, damage from teratogens is not always evident at birth, but may appear later in life. In some cases of malformed limbs, the effect of a teratogen are obvious immediately. Sometimes, however, the damage from a teratogen becomes evident only as the child develops. Have you ever thought of having your own baby? Do you care if it's a boy or if it's a girl, or as long as it's just healthy? In this part, we are going to discuss some of that. Prenatal diagnosis and treatment. Often, the first step in deciding whether a couple's baby is likely to be at risk is genetic counseling. It is where a counselor asks about the family medical history and constructs a family tree for each parent to assess the odds that their child will inherit a disorder. We also have the prenatal diagnosis. After a woman is pregnant, how can we know whether prenatal development is progressing normally? There are prenatal diagnostic techniques that doctors use. One is ultrasound, which is a prenatal diagnostic technique that uses sound waves to generate an image of the fetus. Another is amniocentesis, which is a prenatal diagnostic technique that uses a syringe to withdraw a sample of amniotic fluid through the mother's abdomen. And the chronic villus sampling, or the CVS, which is a prenatal diagnostic technique that involves taking a sample of tissue from the placenta. We also have the NIPT, or the non-invasive prenatal testing. It is a prenatal diagnostic technique that analyzes genetic material released from the placenta that circulates in a pregnant woman's bloodstream. Next is the fetal medicine. A new field called the fetal therapy is concerned with treating prenatal problems before birth. Many tools are now available to solve problems that are detected during pregnancy. We now proceed to the stages of labor. Labor is an appropriate name for childbirth, which is the most intense, prolonged physical effort that humans experience. It is usually divided into three stages shown in the figure. In stage 1, it may last from 12 to 24 hours for a first birth. The uterus starts to contract. The first contractions are weak and irregular. Gradually, they become stronger and more rhythmic, enlarging the cervix. In stage 2, the baby passes through the cervix and enters the vagina. The mother helps push the baby along by contracting muscles in her abdomen. Soon, the top of the baby's head appears or event as crowning. Within an hour, the baby is delivered. Crowning means the appearance of the top of the baby's head during labor. And in stage 3, which lasts only minutes after the mother pushes a few more times to expel the placenta, also called appropriately as afterbirth. The times given for each of the stages are only approximations. The actual times may vary greatly among women. Women who are healthy when they become pregnant usually have a normal pregnancy, labor, and delivery. When women are not healthy or don't receive adequate prenatal care, problems can surface during labor and delivery. Even healthy women can have problems too, but not as often. The more common birth complications are listed in the table. First is the cephalopelvic disproportion, in which the features include the infant's head is larger than the pelvis, making it impossible for the baby to pass through the birth canal. Another is the irregular position. In shoulder presentation, the baby is lying crosswise and the uterus and the shoulder appears first. In breech presentation, the buttocks appear first. Another is the preeclampsia, where a pregnant woman has high blood pressure, protein in her urine, and swelling in her extremities due to fluid retention. And last is the prolapse umbilical cord. The umbilical cord precedes the baby through the birth canal and is squeezed shut, cutting off oxygen to the baby. It is a condition known as hypoxia. It is a birth complication in which the umbilical blood flow is disrupted and the infant does not receive adequate oxygen. In a cesarean section or C-section, an incision is made in the abdomen to remove the baby from the uterus. It is a surgical removal of an infant from the uterus. A C-section is riskier than vaginal delivery for mothers because of increased bleeding and greater danger of infection. A C-section poses little risk for babies, although they are often lethargic briefly from the anesthesia the mother receives before the operation. Problems also arise when babies are born too early or too small. Normally, a baby spends about 38 weeks developing before being born. Babies born before the 36th week are called preterm or premature. In the first year or so, premature infants often lag behind full-term infants in many facets of development. However, by 2 to 3 years of age, such differences have vanished and most premature infants develop normally. Low birth weight has generally a great impact on the baby. Low birth weight means newborns who weigh less than 2,500 grams or 5.5 pounds. Very low birth weight means newborns who weigh less than 1,500 grams or 3.3 pounds. And extremely low birth weight means newborns who weigh less than 1,000 grams or 2.2 pounds. Babies with very or extremely low birth weight do not fare well. Many do not survive, and those who live often lag behind in the development of intellectual and motor skills. For the infant mortality, imagine if you were the proud parent of a newborn and a citizen of Afghanistan. The odds are 1 in 6 that your baby would die before his or her birthday worldwide. Afghanistan has the highest infant mortality rate, defined as the percentage of infants who die before their first birthday. If you're a parent and a citizen of Czech Republic, Iceland, Finland, or Japan, the odds are less than 1 in 300 that your baby would die within a year because these countries have some of the lowest infant mortality rates. In the graph figure 2.13, put these numbers in a global context depicting infant mortality rates from 15 developed nations, as well as for the 15 least developed countries. What explains these differences in infant mortality rates? The United States has more babies with low birth weight than virtually all other developed countries, and we've already seen that low birth weight places an infant at risk. Low birth weight can usually be prevented when a pregnant woman gets regular prenatal care, but many pregnant women in the United States receive inadequate or no prenatal care because they have no health insurance. In the least developed countries, inadequate prenatal care is common, and mothers often have inadequate nutrition. After birth, infants with these countries face the twin challenges of receiving adequate nutrition and avoiding diseases. However, with improved prenatal care and improved health care and nutrition for infants, the global infant mortality has been
And
cut
that
in
is
half
all.
since
Thank you so
1990.
much for listening, everyone.
Low birth weight has generally a great impact on the baby. Low birth weight means newborns who weigh less than 2,500 grams or 5.5 pounds. Very low birth weight means newborns who weigh less than 1,500 grams or 3.3 pounds. And extremely low birth weight means newborns who weigh less than 1,000 grams or 2.2 pounds. Babies with very or extremely low birth weight do not fare well. Many do not survive, and those who live often lag behind in the development of intellectual and motor skills. For the infant mortality, imagine if you were the proud parent of a newborn and a citizen of Afghanistan.